Thanks, everybody. I've got to say it is an honor to be part of this session. The speakers have all been so fantastic and really, really interesting talks, all of them. So I'm going to update you on a team effort to learn what we can from Rice's whale baleen that we've been collecting, or that many people have been collecting from some of the stranded specimens. I'm just one member of this large team, see all the other names here at the bottom, and I hope I do a good job representing everybody. I want to start out by saying that the team overall is looking at aspects of individual whale biology and individual whale decisions, internal decisions of the whale. I'm coming at it myself from a reproduction and stress angle. I've spent a lot of time studying endangered species, critically endangered species, and often I end up focusing on individuals. Why is this individual not breeding? Is this individual stressed? How long have they been stressed? So I really want a close-up look at individuals, and that's really hard to get with the large whales. Other members of our team are studying other aspects, such as exposure to various pollutants and prey selection, which we just heard some really interesting information about. But much of the other members of the team, many of the other members of the team, likewise, are trying to get a really good look at an individual over time. So as a reproduction and stress person myself, what I really want is this view of the same individual whale every day for years, and I want a sample from that whale every day, ideally a blood sample, maybe a poop. I would take poop, but that's the view we want. This is the view we get. The previous talks have been so impressive, but I do want to convey that when the previous speakers are showing these glorious close-up photos and these great tagging data, that work is very hard to do. It's very impressive that it's able to be done at all. And it takes a lot of time and a lot of days at sea, and a lot of days when you can't even get out to sea because of weather and wind conditions. It takes months and months and months to get little specks of data from each individual whale. So again, as a reproduction and stress person, it frustrates me that I can't get to the animals every day, and I really want to see their story day after day after day. And again, I want samples from the animal's body. I want to look inside the animal, see what's going on internally. Why is this animal breeding or not breeding? When are they breeding or not breeding? What happened during the month that we didn't see them? I want all that close-up information. So I've spent a lot of time, I and many, many other people, coming up with a variety of ways to get samples from whales. We cannot get blood samples from living whales. We can't catch them, not non-lethally, not in a way that could get a blood sample. But there are now many other methods for getting physiological samples from whales, and I mean a sample from the body of the animal to tell us what's going on internally. I'll just give you a couple looks at some other sample types that I and many colleagues have spent a lot of time working on. This is a photo of getting a sample of respiratory vapor from a North Atlantic right whale. This was a project I was heading at the New England Aquarium, and we did in fact find we could get respiratory vapor samples, and we can measure the hormones in the vapor samples. I've also spent years and years and years working on whale fecal hormone assays. Originally with the North Atlantic rights. I'm now doing that a lot with gray whales. And as you can see here from this photo, whales do poop. You can get those poops, and they're actually very informative. The particular photo there, again, is a gray whale, in this case, a poop that we spotted from a drone, from an unmanned aerial system. So you can pair drone operators with poop detection and sort of double your bang for your buck. You can also get physiological information from biopsy dart samples fired from crossbows, as we just heard. You get typically with one of these little dart samples a bit of skin and a bit of blubber. The blubber has hormones and contaminants. The skin has DNA and all kinds of other interesting bits of information. So there's been a lot of advances in ways to study these rare and endangered animals and get a look at, again, at the individual, inside the individual. However, even with all these wonderful techniques, which I really love and which I still use and use every day, there's still big gaps in coverage. You get a sample one day, you don't see that animal again for maybe a year, if ever. There's these sort of spotty data sets where you don't really know what happened to the animal in between those sightings. So I was still sort of casting around for a sample type that would let me see every day of the animal. And this is my sample, baleen. So you've heard a lot about baleen. I'm going to give you a bit more of a close-up look at how it grows so that you can understand why I think it's so valuable for giving us, again, these individual stories over time. So this is actually a picture of a Brutus whale, but I picked it because it's a nice photo of the baleen in the whale. So this animal is lunging up out of the water, so the upper jaw is open there at the top. And as you can see from the yellow arrow, there's this fringe of, in this species, whitish material on the right side and another fringe on the left side. That's the baleen. And it's different colors in different species. It's often dark in other species, but sometimes white. So that's the filter feeding apparatus. The whale is taking in a sort of a barn full of water and fish, and it's going to then close its mouth and sort of squish the water out through the baleen, trapping the fish on the inside. So I want to show you a little more about how a piece of baleen is arranged in this overall array of baleen. This particular photo is of a southern right whale. It's a photo from a museum in France that I thought did a nice job of sort of showing how the baleen is arranged relative to the skull. So you can see there's, again, this long sheet, very long in right whales, long sheet of baleen on the right side and another long sheet or curtain on the left side. Those are called racks. So there's a right rack and a left rack. And each rack, I think you can see in this photo, is made up of a whole series of skinny little pieces. And those little pieces are called plates, and they're made of keratin, the same keratin, same type of keratin you have in your hair and your fingernails. So it's keratin. It's rather like sort of a blend of hair and fingernail that's growing slowly from the whale's upper jaw. So each plate of keratin, each plate of baleen, is shaped kind of like a long, skinny isosceles triangle. And what's really going on is it's growing slowly and steadily from the upper palate, which we call the base, even though it's the upper side of the baleen. That's where it's anchored. It's growing from the upper palate and slowly extending downward. And as it's used in filter feeding, it erodes away down to this skinny little tip, and then the tip is eroding off. So there's eventually in an adult whale a steady state where the baleen is growing at a certain rate at the base, and it's wearing away at that same rate at the tip, and you've got sort of this constant length of that plate of baleen, but it's constantly kind of replenishing itself from the base. And it's incorporating information from the animal's blood while it's growing. It's laying down hormones and stable isotopes and contaminants and DNA, all sorts of other goodies, day after day after day. So there's our time series. There's our how do we look at an animal every single day. So this right whale has quite long baleen. Rights and bowheads have the longest baleen, the bowhead especially. Rice's whales have pretty short baleen. It's a lot shorter than shown in this big right whale mount. So there's one piece of Rice's baleen. And they're just, basically a foot or two. So they won't have as much time captured in them. Their time frame is going to be shorter, but I still thought it would be worth looking at. And multiple colleagues also had the same idea, and we all teamed up. Before I go on and show you a little more about what we've managed to learn with the Rice's whales, I want to convince you that baleen is telling the truth. So I'm going to show you one of the first data sets that I and other colleagues managed to get on baleen from an animal whose history we already knew from sightings, so that we can compare what we knew her history was to what the baleen seemed to be saying. So this is, I'm going to be showing you data from a North Atlantic right whale. Her name was Stumpy. You've actually heard about her a couple times. And any of you who went out to the ocean hall would have seen, I think, the big mount of punctuation, the North Atlantic right. She was Stumpy's daughter, and there is actually a piece of Stumpy's baleen in the ocean hall, kind of off on the side, on the left side, as you go into that hall. So I had another piece of Stumpy's baleen that was on loan to me from Michael Moore, who you heard talk earlier. And so I've laid this diagram out as if Stumpy's baleen is sort of shown there in blue at the bottom. So the base, the newest grown baleen, is on the right. The tip is way over on the left. That's the baleen that was grown a while ago. And I teamed up with Nadine Lasiak, who you saw in a little video earlier that Michael Moore showed of drilling some baleen. Nadine had already done stable isotope analyses on Stumpy's baleen for her PhD. And she was able to determine, I won't go into the details of the isotope analyses right now, other than just to tell you that Nadine had figured out the baleen growth rate of Stumpy's baleen specifically, and of North Atlantic right whales generally. So with that growth rate, we were able to look at that baleen plate and say, okay, this spot on the plate, which was 25 centimeters from the base, say, we knew the growth rate. We knew then when that spot had grown. And we were able to assign a timeline to this baleen plate so that every point on the plate corresponded to a certain year and season. So I've shown that timeline there below the big gray box as the estimated date of growth. And then we had a sightings record for Stumpy. We knew when she'd been seen with a calf and when she'd not come to the calving grounds and had not been seen with a calf. So the black dots in that little calf seen, not seen area right below the timeline, the black dots are times she was sighted not on the calving grounds, not with a calf. Look at the green dot. That is when she was spotted with a newborn calf on the calving grounds. So we knew she'd been spotted late 96 with a calf. We knew she'd been pregnant then the year before, during the calendar year of 1996. Look at the little green triangle way on the right side. She was unfortunately hit by a ship and died from ship strike in early 2004. She had a full-term fetus inside of her, which was quite a tragedy. But that gave us one other known pregnancy. We knew she was about to give birth because of the size of the fetus and where she was. She was almost at the calving grounds. So we knew she'd been pregnant in 2003. So I had the opportunity to get this baleen plate in my hands, and I drilled powder from it every 4 centimeters and basically mixed it with methanol, which is very good at extracting hormones out of dried powdered samples. And I assayed the resulting little extracts for progesterone, which is the major pregnancy hormone in mammals. And I will finally show you Stumpy's data. This was the first time series data set that we got on any hormone for any whale. And the yellow bars show her known pregnancies that we knew from her sightings record, and look at how her baleen progesterone, which is the black dots, which is on a log scale, look at that y-axis, went incredibly high during those known pregnancies and then came very low between the pregnancies. So we knew then the baleen was telling the truth. It was telling us when she really had been pregnant. And side note, it also told us that she had not gotten pregnant in between those two. We'd wondered why, if she'd had a calf that died, but no, she just didn't even get pregnant in between. So I spent a while talking you through that so that you'd understand the graphs I'm about to show you. They're all going to be laid out this way with time going from left to right. And with hormone shown. But again, right whales have really long baleen. Rhesus whales have much shorter baleen. We thought we might only get a year or two, if we were lucky, out of the Rhesus baleen. So we weren't sure how much this would tell us, but we thought it was worth a try. So with all that as background, I'll take you into our Rhesus whale baleen project that is currently framed up. So we've got multiple different teams looking at different aspects of the individual whale's biology. I'm on the hormone team, and so is my PhD student, Rebecca Evie, who's in the audience with us here today, and I'm going to be showing some of her data. And some of our major major questions with the hormones are, could we tell if a female was pregnant? And we're going to be looking at progesterone to determine that. We can look at testosterone in males too, and male whales that have regular breeding seasons have a seasonal elevation of testosterone at that time of year. So we were curious if Rhesus would show anything like that. And I was very interested, I and Becca both very interested in the patterns in stress hormones. And there's actually multiple stress hormones. I'm just going to show you 2 that happen to be named corticosterone and cortisol. Another team is looking at stable isotopes of the baleen. So again, the baleen goes slowly over time. You just heard, I'm very grateful to Jeremy for explaining stable isotopes, because you just heard that analysis of stable isotopes can tell you what prey a whale has been eating. And now I think if you picture that timeline of the baleen, you realize we can see what prey the whale was eating over time, day by day by day, over maybe a year and a half from the Rhesus whale baleen. If the whale switches prey, that sometimes will tell you that the whale has moved, and that can tell you sometimes if the whale is migratory or not, if that species is migratory. And we were suspecting these guys weren't, but we thought it would be interesting to look at it. We also have a contaminant team. We don't have data in yet from the contaminant analyses, but just so you know, you also can look at contaminant load, and you're looking at the circulating contaminants. The baleen is a proxy of the blood, and you're looking at the contaminants that were circulating in the blood at different points in time. Again, I'm just one member of this vast team, so it was, of course, John Osowski and Michael McGowan who were able to give us access to the specimens of Rice's whale baleen. Everything I'm going to show you is from specimens that the Smithsonian was curating, including some old ones from the past that we now realize were probably Rice's whales. Matthew Savoca, Dr. Savoca, is the fellow who really had conceived this whole project and pulled the whole team together, and he's really leading the whole thing. And I think he's watching online, so hi, Matt. Hope you're enjoying the talk. And then there's multiple other teams, as I've mentioned. Myself and Rebecca Evie are on the hormone team. So I don't have time to show you all our data, especially since I've spent so long explaining baleen, but it always has to go like that because I need you guys to understand the timeline. But just to tell you what our overall data set is, we've got 7 specimens of baleen from 7 different Rice's whales that the Smithsonian had that we were given sampling access to, which is amazing. I'm just going to show you bits of data from the adult female, who you've already heard about. One little extra detail on her, the necropsy showed she was probably lactating when she was struck by the ship and killed. So she should have had a prior pregnancy. And I'll show you a little bit of data from the holotype, who you've heard quite a lot about. This is the animal killed by a piece of plastic. Here is what baleen looks like in a Rice's whale. You've seen a couple of photos of this already, so much shorter than right whales or bowheads, but there's still some months of information in there. So for sampling baleen, let's see if I can get this to run. Matt has, there we go. This is Matt Savoca, who did all the sampling mid-pandemic. It was difficult to get into the archives during this time, but Matt managed to get approval. And what we do is very carefully measure centimeters from the base, and we mark all these little sampling spots that are carefully measured along the length of the baleen. And then we use, as you can see, a handheld Dremel. And those of us who do baleen work have gotten very good at Dremeling things precisely and making precise little grooves. And Matt is collecting the powder and then dividing it up into different vials for the different teams. One of those vials came to me and Becca. So let me show you the adult female's progesterone. Again, this is an animal who we think was lactating when she died. And again, just reminding you of what the graph is going to show. The x-axis is, it's shown here in centimeters from base, but it's really a timeline going from left to right. She dies on the rightmost end, and we think we've got about 1.5 years total. So the left side is 1.5 years before she died. And then I'm going to first show you progesterone. And there's her progesterone. So again, this is the pregnancy hormone, and this is a classic pregnancy profile. I think we caught her full pregnancy laid out here in that piece of baleen. So she was, we think, pregnant, and we can see the initiation of the pregnancy and as it went toward term. Now, we knew at some point she probably gave birth because she was lactating when she was killed by the ship. So here are her 2 stress hormones, corticosterone and cortisol, and you can see they both start going up toward the end of the pregnancy. That's normal for mammalian pregnancy. Those both normally do go up in late pregnancy. Pregnancy is a type of a stressor. But look at corticosterone doing that huge jump. So corticosterone often does that at birth. In fact, corticosterone often is one of the signals that triggers labor. So we may have captured the parturition event of the calf in that profile of the 2 glucocorticoids. That's the name for corticosterone and cortisol together. From this female, again, we only have 7 pieces of baleen, and I only had 1 lactating female, but it does look like we are able to say something about the animal's previous year. That is the truth. Here is I'm just going to show you the stress hormones of the holotype. I'll put them up. You know now how these graphs work. Both his stress hormones take this big jump at the end, and the ones I've outlined in boxes are indeed significantly higher than the baseline levels of those hormones for that whale. This is the animal that died from the piece of plastic rattling around in his stomach, and I think we're seeing here the stress of about 1 to 2 months before death. So this one, unfortunately, looks like it may have been a prolonged stress event for this whale. The last bit of hormone data I want to show you is testosterone. So we've got 3 males, which is not many, but to have 3 adult males at all is 3 more than we've ever had. I first want to show you what what a typical whale looks like. So on the left, I have testosterone profiles over about 1.5 years from 5 bowhead whales. So bowheads are typical whales in that they are migratory and they have a very well-defined breeding season. They all breed at the same time. And in species like that, as you can see here, testosterone in males goes up at a certain time in year, and you can see that in the baleen, and then it comes back down again when breeding season is over, and it goes up simultaneously in all the males in the same months. So that's what a seasonally breeding whale looks like. Here's our 3 adult male rhesus whales, and you can see there's jitter. T does go up and down a little bit, T, testosterone, but it's not anything, I don't see any regular annual cycle there, and the 3 individuals are not synchronized with each other. So I used to do a lot of work on seasonal and non-seasonal vertebrates, not marine mammals, but other vertebrates. For instance, I did a lot of work on seasonal and subtropical birds. And if I didn't know these were whales, if someone just showed me these 2 graphs, I would have said the one on the left looks like an Arctic bird, and the one on the right looks like a Panama bird. This is a classic. This rhesus whale graph on the right is classic for a subtropical species. I will add that doesn't mean they don't have a breeding season. Often the subtropical vertebrates do have a time of year when they're more likely to breed. They often will have a breeding season, but it's longer and it's more flexible. The whales are better able, or the animals are often better able to say, like, I could breed this month, but conditions aren't perfect. I'll wait till next month. They're more likely to delay reproduction because they have this longer breeding season and they can get away with doing that. So that's what I have to show you for today for hormones. We have many more profiles from the other individuals, but in the interest of time, I'll start wrapping it up. I do want to give you one quick peek at the stable isotope data. This is the ratio of the rare nitrogen isotope to the common nitrogen isotope. And long story short, these are all the 7 whales over about 1.5 years, sometimes less for the younger individuals. Look at whether the lines are showing a similar pattern, and they're really not. So really, much like the testosterone, there's no clear annual up and down, and they're not showing the same pattern. This is, again, typical of animals that are non-migratory and that are not doing a lot of prey switching. I'll point out one other thing quickly before I wrap it up for the data. Look at the blue line in the middle panel, in the middle row. See how high it is? That's an animal that's feeding at a higher trophic level. That was a calf. It was feeding on milk. That's a higher trophic level. It's kind of like it's eating its mom in a way. So calves typically will have that higher line, and that indicates lactation. Now look at the upper right. That was a subadult female. And I saw her isotope, I first saw her hormone pattern. I haven't showed you her hormones, but there's a lactation signature of hormones that I don't have time to show you, but I'll just say I saw her hormones and I thought, looks kind of like she was nursing in the first half of her baleen plate. I mentioned this to Matt Savoca, who had this isotope data in front of him, and he said, look at the comment I already put on that upper right. And I clicked on the comment, and it said, nursing? ? So the isotope and hormone teams both independently picked out that subadult female as having probably been nursing on milk a year earlier, and we think we caught her weaning event. So this might be informative for telling us something about when do they wean. Subadults are also useful for informing us about age of sexual maturity as well. So that's all the data I have to show you today. I'll wrap up because I'm out of time, but just to run through some conclusions. We can detect prior pregnancies in baleen. We're not seeing any evidence of a distinct synchronized breeding season, which kind of makes sense for this subtropical whale. We are seeing stress hormones go up when there's prolonged chronic stress, so that might help us figure out cause of death, or at least how long the event lasted. No evidence for prey switching. We think we're picking up a nursing signature. More results coming soon. There's other analyses that are still underway. It's only 7 individuals, but we have this lovely time series for each individual. I always find it so tragic when a whale dies, and I feel this responsibility to learn as much as we can from each such event. So we're going to be learning everything we can from each of these precious pieces of baleen. I want to thank everybody. There's many, many funders, and I especially, of course, want to thank John Osowski and Michael McGowan and the Smithsonian for giving us access to the specimens. It's been so critical to be able to get our hands on those specimens. I'll wrap it up there and take any questions. Thank you very much.
Thanks so much, Kathleen, for the amazing data profiles. Do we have time for like 1 or 2 quick questions? I see one person.
Yeah, so I can't remember when the 2009 female stranded, but roughly how many months back was the big spike in the stress hormone that you think indicated, like right when it gave birth?
We're guessing a little bit about baleen growth rate in this individual. So they don't have clear annual stable isotope cycles, and that makes it harder for us to figure out baleen growth rate of each individual. But I have found over my last many years of doing a lot of baleen work that a lot of adult males actually have a bit more of there's a more consistent baleen growth rate in all the adult masticetes than I originally was expecting. And we're thinking it was probably only about a month before, but I got to hedge that with a lot of, we don't know her baleen growth rate, and we're also not totally sure what the temporal resolution is of 1 point on the baleen. Is that a day? Is it an average of the surrounding month? We're not quite sure about that yet either. About a month is my best guess.
Okay, thank you.
Sure. Any other questions? Do we have time? Any questions? All right, thank you very much.